that were not completed as little snippets that can be posted. And I am expecting a more normal life, and after all the events that I've had to get through in the past 6 months. So we should be able to keep pace with this. I appreciate Marco's presentation on Friday. I watched that this morning before leaving for lecture. Seemed to go quite well. So we'll pick up with the digestive system and move on. We have seen much of the digestive system form already in using that as a reference point for other related structures, especially the pharyngeal pouches as a prime example. But we begin first just looking at the foregut, there should be an E in here, sorry, and the mesenteries associated with it. So we've already discussed this issue of the gut tube and mesentery with the parietal and visceral layers, the two balloons coming together to squash onto a pencil, and we will build on that model through today's lecture, and it will be our model throughout the discussion of the gut tube. And we'll see how it gets twisted and contorted, and some of the ventral mesentery in front of the pencil where the two balloons make contact is going to persist. Some will disappear, and we'll talk about what there is dorsal to that pencil on the back side, excuse me, that mesentery carrying the vasculature, the neurovascular bundle, and what happens to it. So we start first reminding ourselves, what are the derivatives of the foregut? Well, the pharyngeal gut, as we had seen already, the tracheobronchial diverticulum, that it was involved in the discussion of the development of the respiratory system, the esophagus, or esophagus, the stomach, the upper part of the duodenum, the gallbladder, pancreas, and liver. So these are the structures that we're going to be discussing in today's lecture, and there'll be a bit more detail when we talk about the development of the accessory glands as we proceed. So the normal adult is a good starting point. We will take a quick look again at the oral cavity, then the pharynx, the esophagus, the stomach with its curvatures, and the small and the large intestines, and then those accessory organs that we just saw on the list, the pancreas, liver, gallbladder, and the spleen. Our first slide of the adult structures is shown here. We remind ourselves of the oral cavity with the oropharynx, the nasopharynx above, the laryngopharynx below, and now we are taking our route through the esophagus that is posterior to the trachea that developed in this area from the laryngotracheal groove and bud and so on and so forth, the lung bud that becomes the bronchial bud. I will make a point of emphasis as we do proceed, but I noted in Marco's lecture discussion on the distinction between mesoderm and mesenchyme, and it is a bit blurred. It does, in general, follow the sequence of terminology change in representation of a further lineage of commitment to differentiation. We've talked about this from totipotent to pluripotent to multipotent to tetrapotent to bipotent and unipotent, all the way down through. And indeed, mesoderm does pick up this term mesenchyme as it has achieved a later stage of development and has migratory patterns and a variety of things like that. I was specifically continuing to use the emphasis of mesenchyme in the development of the face, of the pharyngeal arch regions, to remind us that when we saw mesenchyme there, we thought immediately of neural ectoderm giving rise to that mesenchyme, that specialized mesenchyme. Mesenchyme from here on out will pretty much be referring to mesoderm, the typical third layer, middle layer of the germ cell, 3 germ cell layers, giving rise to anything we call mesenchyme, basically from here on out, basically again from this point of the esophagus on down through. So I had allowed it to be a bit misleading up to this point, primarily so that we associated mesenchyme with pharyngeal region with a specialized origin, the neural crest cells, and we mentioned a mixture of those mesenchyme from neural crest cells with some of the mesoderm contribution in the development of the pharyngeal arches. Now, basically, we'll be talking about mesenchyme of typical middle layer mesodermal origin. So we'll bring that into play as we talk about the different layers of the esophagus and the gut tube in general, and the origin of each of the different layers. Then, of course, the stomach, and we can see that there's one side of the stomach that now faces the right-hand side. It's the original surface that faced the front side, the anterior side of this individual, and then one that has a very elongated curvature, so the lesser curvature and greater curvature, obviously. Excuse me. That used to be the posterior side, but because it got so long, it couldn't stick posteriorly anymore, so it got tossed over to the left-hand side, displaced there primarily by the liver growing within the mesenchyme, if you will, of this area of the upper right abdomen. So we're beginning to make these associations at the adult level, and this reinforces the statement that Marco made as well, that if you can envision the adult organization and structural features, then you're trying to follow development from a simple linear tube, in this case, into something that looks like what you can picture from the adult, because we've seen it so many times in our education process. We remember that the gallbladder is part of a duct and tube system that goes all the way into the liver and connects to the duodenum. So we'll differentiate the origin of the different pieces and parts of the biliary tree, including the gallbladder, versus the liver itself. And we'll follow down into the duodenum. So after the stomach is the duodenum, and the pancreas is right here, as we'll see in the hugging arms of the duodenum. Then later lectures will go through the small intestine and large intestine, differentiating foregut, midgut, hindgut contributions. But first, we'll go back again to the oral cavity, reminding ourselves of the development from the tip of the uvula, the soft palate, to the pretty much the base of the tongue, where the sulcus limitans, a V shape that ends in the foramen cecum, would be positioned. If we draw a line down here, we see that that's where the original buccopharyngeal or oropharyngeal membrane was located. So this is all surface ectodermal origin in the oral cavity, all up along the lip, or the labia inferia and the labia superia, on out to the external surface of the body. Whereas the nasal cavity, it's up in this area here, just inside the nostril, all the way back to the posterior choana, excuse me, and into the nasopharynx, where we find the opening of the pharyngotympanic tube, the auditory tube, the eustachian tube, a variety of terms for it, that opening in this area. So we're recalling these were pouches and things like that. So this is all endodermal origin out to this line in this area. So there's hair follicles in that zone. And then the oropharynx, and then the laryngopharynx, and it was the laryngopharynx where we saw all the structures, hypobranchial eminence, contributing to the formation of the epiglottis and its cartilage, and then the laryngotracheal groove, developing into all of these structures here for the larynx. Now we start with the esophagus as it proceeds posterior from this point, and that will be where we pick it up. This is not an illustration of the esophagus. We're using it as a general composite image of the gut tube in general. And the reason for that is it shows a variety of components that we don't find every location along the gut tube. For example, the esophagus is not going to be the exact same as the stomach wall or the duodenal wall or the colon wall. So this just allows us to illustrate the three general layers. We see first over on the right-hand side the mucosa, the submucosa, the muscularis externa, and the serosa, the four layers. Excuse me, I believe I might have said three. So there are these four layers present in the gut tube. We start with the middle, so if you're standing in the lumen and you begin to pass through, or if an object has been swallowed and is now beginning to pierce the gut wall from the inside out, it would first go through the epithelium of the mucosa. Next, there's an underlying loose connective tissue called the lamina propria, and then there's a thin layer of smooth muscle called the muscularis mucosa. So the mucosa of the gut tube has three linings, three layers: epithelium, lamina propria, muscularis mucosa. Epithelium is a lining cell, the layer of cells on the lining of a tube or on the surface with the external environment, and this indeed is the external environment. The lining, the epithelial lining of the digestive system, from that point that we just mentioned, from the soft palate down to the sulcus limitans and the foramen cecum, is originating from the endoderm. As well, the endoderm pushes down into the underlying mesodermal layers and will develop into glands. So here we see a gland in the mucosa. Here we see one going very deeply into the submucosa, passing the muscularis mucosa and forming the glandular portion and the persistent and patent duct opening onto the surface of the lumen of the digestive tube. Then on this portion over here, we see the duct of a gland that will end up outside, for example, pancreas or even gallbladder. So the same thing, the endodermal cells will push out into the mesoderm and continue to grow outside of the tube that is forming. So there are tremendous communications between cells as these developmental events are occurring, such that the splanchnic mesoderm, the intraembryonic splanchnic mesoderm, is going to differentiate into the connective tissue, the smooth musculature, or all musculature, connective tissue as well. We see arteries and veins, the red and the blue, the vasculature. We see over here neurovascular bundles. As soon as we hear a neurovascular bundle, we look for what's missing, and classically what's left out is the lymphatics. So the splanchnic mesoderm in this area is differentiating into veins, arteries, and their associated structures, the capillary beds, venules, arterioles, and so forth, as well as the lymphatic vessels that would be accompanying them, and then the lymph nodes as well. Not the cells inside the lymph nodes necessarily, but the structure of the lymph node itself, as it's just kind of a little dilation and housing created along the length of a excuse me, of a lymph vessel. The nerves, we are reminded, sensory or peripheral sympathetic, postganglionic sympathetic. There's postganglionic parasympathetic neuron cell bodies in the submucosal layer and in the muscular layer, not shown here, that differentiate from neural crest cells. All peripheral neurons, especially when the soma and the dendrites and the axon are all contained within the peripheral nervous system, come from the neural crest cells. But any other type, well, we wouldn't see it here. Well, there would be a preganglionic parasympathetic axon, coming from typically the vagus nerve, so it would be the brainstem, the medulla oblongata. There's still a central component that originated from the neural tube neuron who sent its axon out, and it got stretched all the way out through. So all these things should come to us whenever I point to a neuron, and I know I throw them at you every time, but as the semester continues, I'll stop doing that, and it should flush through your mind every time you see a neurovascular bundle. All these facts should emerge from your long-term memory. You must put forth that energy to make this working knowledge. So endodermal origin, producing the glands. All the other is going to be the splanchnic mesoderm because it's all connective tissue, excluding nervous tissue within here. It's all connective tissue, vasculature, and smooth musculature. Same with the serosa. It is a connective tissue with a lining epithelium. So we remind ourselves of the layers again: mucosa, submucosa, muscularis externa, and the serosa. So the mucosa we've discussed quite thoroughly. The submucosa is a connective tissue, a loose connective tissue. It basically provides a housing for the neurovascular bundle and the glands if they are present. It's also a cushioning in this area, allowing stretching and compression as boluses of food or digested, partially digested. There's other terms for it. Chyme, for example, in the small intestine is being propulsed through. It is going to cause stretching and compression in these areas. The muscularis externa has two layers, inner circular and outer longitudinal. And then the serosa is called a serosa only if there is a visceral peritoneum lining the surface. So we've talked about the formation of the visceral and parietal peritoneum, and where they attach and communicate with each other, being a mesentery, and we will continue with the discussion of that. But now we can envision all the layers of the gut tube. That layering is present anywhere. I need one more point before I leave that slide. There will be some places where there will not be a surface layer of epithelial cells. For example, the esophagus and the posterior mediastinum, the posterior thoracic cavity. There is just the surrounding loose connective tissue is continuous with the loose connective tissue in the area. There is no surface epithelium of simple squamous cells, so it's called an adventitia. So ADVENTITIA, adventitia, is the serosa without the lining cells, if you want to think of it that way. So the primordial gut develops during the 4th week. So we'll see the 3-week embryo as we have seen all along. We've already had sufficient cranial folding and caudal folding. So by the 4th week, we've got a significant, at least initial, constriction of the body stalk. And when we get that constriction of the body stalk with head fold, tail fold, lateral folds, then we know we've begun to incorporate some of the yolk sac into the fore region and the hind region, and we have foregut, midgut, hindgut. And this is where this slide is taking us. Oropharyngeal membrane, we talked about where it's located, the soft palate to the sulcus limitans of the tongue. Cloacal membrane is where the proctodeal plate, I don't believe I'm using the correct term there, apologies, was located, as we saw, early in the embryo. So the prochordal plate became the oropharyngeal membrane, and then the one at the other end, I'll not use the mistaken term, becomes ultimately the cloacal membrane. See, if you don't call it back and use it all the time, it doesn't become working knowledge. So the foregut then will include the pharynx, the oral cavity, the glands in this area, the respiratory system, the esophagus, the stomach, the proximal duodenum, the liver, the gallbladder, and the pancreas. These are all derivatives of the foregut. The midgut will have derivatives that are the distal duodenum, the rest of the small intestine, and the proximal large intestine up to the transverse intestine where it comes across just under our rib cage. And then the hindgut will be the distal large intestine and the epithelium of the bladder and the ureters. So we'll take this discussion right into the development of the urinary system at some point. So here we see the 3-week embryo from a lateral view. We're seeing the head fold with the brain vesicle developing here and the tail fold, and of course with the neural tube being formed and closed here posteriorly as well. Excuse me. And we see that the gut is cut along its edge, and we can follow that margin. And we note the body stalk here in the anterior or ventral midline of or midpoint of the embryo. And if we can draw a virtual line that goes right along this one here where they're pointing to the site of the liver development, this is the junction of foregut with the midgut all the way back where we bring this same type of a horizontal line in here, and the yolk stalk ends, then that's the junction of midgut and hindgut. We already see the cloacal membrane and the oropharyngeal membrane being present at either end of the gut tube. And we'll continue on with the relevant discussions as we proceed. This slide is calling to our attention the fact that vasculature has a key association with and a key interaction with the organogenesis. There is signaling. There are signals that tell mesenchymal cells to continue or complete their lineage of differentiation to become vasculature. So angiogenic triggers are emerging from the cellular interactions of all of these structures. We saw that the arches contained branches of the aorta. So major arteries were contained within there, and we'll see what those develop into later. We now see that there's a branch from the dorsal aorta for the foregut, a branch for the midgut, and a branch for the hindgut. So the celiac trunk, actually, or celiac artery, is the supply for the foregut. There's some minor exceptions to this because of the foregut continuing all the way up into the oral cavity, but that's fine. Once we get to the thoracoabdominal diaphragm, these association of arteries with the portions of the gut tube stand true. So the celiac artery will provide arterial supply to the abdominal esophagus. There's different origins of arterial supply to the thoracic esophagus. So let's just zip past that and continue with this generalization. Once we're in the abdomen, celiac artery is foregut. Superior mesenteric artery is going to provide arterial supply to the midgut. Inferior mesenteric artery to the hindgut. So these types of associations are extremely helpful when you're studying adult human anatomy and you are asked what is the arterial source to this portion of the gut tube. If you know that portion of the gut tube is a derivative of the hindgut, you know that there must be, or is very likely to be, an arterial supply coming straight off the inferior mesenteric artery. A lot of branches in between, but you at least can get to that association. So we see that the hindgut is developing into the cloaca with the cloacal membrane, and the proctodeum is on its way to being developed into the anus. So if you want to be somewhat polite to someone, but being still just a little nasty, you can call them a proctodeum, and that they're well on their way to being an anus. So that's even worse than the pilot's jokes that telling flying us home from San Diego yesterday, saying there's broken clouds over Houston. I don't know how they're going to fix them, but they called together a committee, and they didn't come up with any way to fix those broken clouds. Okay, so let's move on. Here, obviously, is an illustration that you should be able to label. We've seen it once before, and I'll leave you on your time to make the mental associations. The correct responses are given here for one, obviously, as an example, the pharyngeal gut, the pharynx. Up here, two, tracheobronchial diverticulum, another term for, we've used that term previously. So I'll let you work your way through and take a look at that. It's obvious examination material. So you should be prepared to identify all the structures on that by the time of exam 3. So the esophagus develops from the partitioning of the laryngotracheal groove. We've mentioned this. No surprise to us, nothing new there. That tracheoesophageal septum that had to form completely to separate the respiratory from the digestive system. By week 7, it will reach its final length. So the esophagus is going to lengthen as the thorax lengthens, as the neck lengthens. We've referred to that repeatedly. We know the heart pulls down in from in front of the head, and basically the heart bulge occupies the future thoracic region, and the heart is going to have to become relatively smaller than everything else that grows in this area. There has to be a tremendous lengthening and increase in the dimensions all the way around of the thoracic region. So there is a final length, but it's still quite early on. It's 1 week before the fetal stage. So the epithelium and glands are derived from the endoderm. And looking at the model of the adult structure, we saw how that occurs. There's a rapid epithelial proliferation that closes the tube, and then it reopens by week 9. Anytime we hear about this occurring, where it either starts as a solid core and has to become patent, or if it's a hollow tube, and then there's an epithelial proliferation that closes it off, we know that it has to reopen, and there can be problems with that. We don't reopen the esophagus. There's obviously going to be problems in swallowing, and we have problems with the eating of the newborn, and we have to remediate that as soon as possible. And of course, typically it's going to require some type of surgery. So the skeletal muscle will be found at the upper 2/3 of the esophagus. So the cells of origin, mesenchymal cells. So mesenchymal cells, mesodermal cells, or neural crest cells, depending upon where we are, we're up pretty high. So these skeletal muscles are likely to have originated from mesenchyme of neural ectoderm, neural crest cell origin. And then finally, the distal 1/3 will be smooth muscle, and that is from mesenchyme of splanchnic mesoderm origin. So now we see we can reemphasize this distinction. It's all mesenchyme, but when we're up in the pharyngeal arch region, it's more likely to be mesenchyme of neural crest cell origin. As we proceed away from there, it's more likely to be, and definitely will be, mesenchyme of mesodermal, classical mesodermal cell origin. Excuse me. So, in referring to the adult structure, we know that if we swallow something and we just barely kind of get it down into our throat, as we call it, or into the esophagus, we can work to get it back up. But once it hits a certain point, you can't do that. It triggers the involuntary nervous reflex response to produce the peristalsis, as we know is the term for that muscular reaction that moves the bolus of material from oral to the anal direction through the digestive system. So we know there's skeletal muscle. Now we can actually realize it. We know we have voluntary control of the upper part of the esophagus and pharynx, but once it hits a certain point, that junction of 2/3 to 1/3, boom, it's gone, it's down. So the muscle that innervates this area is cranial nerve 10, the vagus nerve. So this nerve jumps on the foregut when the foregut is still pretty short, and as the foregut grows and lengthens, then all the axons grow in length as well, the axons and the peripheral processes of the sensory neuron. So this is a histological slide. Both are really of the same type of a stain. Actually, they are a bit different in stain. We see in the lower panel, the nuclei are of a blue color, and there's a more pinkish to blue coloration of the cytoplasm. Then in this upper left panel, everything is basically red, so it's some type of a red dye that was used, but that's fine. What we notice is that there's a surface in the upper panel. This would be the lumen. And then we go deeper and deeper and deeper, and we see the cells take on different shapes as we go from the lumen to the deepest part. The lower left panel shows us the deepest part of the surface epithelium. So the endoderm would terminate here, except where it might have pushed in to produce a gland. We don't see that at this point. And then just underneath would be the lamina propria. There isn't really a distinctive muscularis interna for the esophagus. Then the submucosa would be in this area here, and then this is the muscularis externa, and then there'd be an adventitia, connective tissue, without surface epithelium on the outside. So we go back to the surface lining. We see that the cells lining the surface have nuclei in them. They are alive. So this means that this is a stratified squamous epithelium, and it is non-keratinizing. That's classical of the oral cavity, the pharynx, and the esophagus. Here we see the musculature, and I noticed from looking at this that this is skeletal musculature, so it must be the upper 1/3 of the esophagus. You don't need to be that good at it, but you do need to recognize that this is the esophagus. The germ cell origin, if we were to point to these cells here, would be endoderm. If we were to point to the skeletal muscle cells over here in the muscularis externa, these would be the mesenchymal or mesodermal or splanchnic mesodermal cells of origin. And the innervation, of course, is cranial nerve 10, the vagus. So the stomach is next, and its coverings. It starts as a simple linear tube. The posterior portion is going to grow faster and become the greater curvature, and it's going to get thrown over to the left because of the liver growing over on the right side. So we saw that in the adult picture earlier, so we'll push right on through. We've seen with our black and white line drawing the stomach up here in this portion of the foregut, just distal or caudal to the area where the laryngotracheal diverticulum had formed. We see the celiac trunk, the superior mesenteric and the inferior mesenteric arteries, the yolk stalk, giving us a line of separation of foregut from midgut from hindgut. We recall the allantoic stalk and the yolk stalk are going to become incorporated into the umbilical cord. So we keep those terms in mind. The cloaca, the cloacal membrane, and then our friend, the almost anus, the proctodeum. So the stomadeum and the proctodeum are terms that we continue to use. So we had talked about the lateral folding, and we talked about two balloons coming down on top of a tube, a straw in the center. And we looked at that, and we did a cross-section. I will call up my pencil or the pen on here and illustrate that again. So in cross-section, we saw one balloon on one side that came up and surrounded, and then one balloon on another side that came up and surrounded. Surrounded a tube with, tube has, this is an epithelial lining here. So this outer portion would be the serosa, external, or muscularis externa, the submucosal layer, and then the mucosal layer. So we know the layers of this structure right in this area now that were just created from this lateral folding of things collapsing, the two balloons, and we know that they are the pericardio-peritoneal canals, those ventral-most or caudal-most extensions, both of which collapse down upon the gut tube as the yolk sac is drawn within to the embryo. So where the two layers are together dorsally, this is dorsal here, that's the dorsal mesentery. We see that on this illustration here. The dorsal mesentery goes back to the dorsal aorta, so this dorsal mesentery carries the neurovascular bundle, and there we see the foregut artery, the celiac trunk, coming to this localized dilation, the primitive stomach. The ventral mesentery continues toward the anterior body wall, as this would here. It will not always persist. Some things will grow into it, like the liver, and we'll see all of this as we proceed. But for now, we're focusing on the stomach. So we have the two balloons collapsing on it, forming a dorsal mesentery of the stomach. The dorsal mesentery of the stomach is called the dorsal mesogastrium. So gastra, we know, means stomach, and IUM just means structure. So the dorsal mesogastrium is the dorsal mesentery of the stomach. And then this other is the ventral mesogastrium, continuing on down through. So we're going to use this terminology as we proceed. Dorsal mesogastrium, ventral mesogastrium. Dorsal mesentery proper for all the midgut portions, and the ventral mesentery proper, and the dorsal mesocolon and the ventral mesocolon. So we're going to use that terminology throughout. We envision a linear tube with this two balloons coming onto it and forming that all. Then it's going to grow and twist and contort and change tremendously. So start with this simple vision in our mind, and we'll proceed from there to follow these changes. The dorsal mesogastrium undergoes tremendous lengthening, and the dorsal surface of the stomach also grows much more rapidly in its rostral to caudal dimension, linear dimension, than does the ventral aspect of the stomach. It becomes the greater curvature and the lesser curvature, respectively. The dorsal mesogastrium is attached to the greater curvature of the stomach, and it elongates and is pushed off to the left. Some structures develop within there, ultimately the pancreas and the spleen, and we'll follow those at a later date. The celiac trunk or artery stays within there. We're going to see a dilation of this, a narrowing of the dorsal mesogastrium to be just a small opening, as we see in the next slide, and a little pouch forms over here, and it's called the omental bursa, and it will be referred to as the lesser omental bursa, a fluid-filled sac or space, and then all the rest of the peritoneal cavity is going to be called the greater omental bursa. So the lesser omental bursa is shown with this broken line in here. So if we were standing in this little hole, the omental foramen, and looking in, it would be like looking in a purse where there's a purse string that puckers it up. You'd be looking in and seeing a dilated area of fluid-filled space. There's nothing in there. It's a potential anatomical space. It's all squashed down and just a little bit of fluid in there, as within all of these pleural cavities, peritoneal cavities, and pericardial cavities. Potential anatomical space, the thin film, so we get a nice smooth movement between the epithelial layers, those simple squamous epithelial cells. As Marco mentioned before, in any one of these cavities, you injure those epithelial cells. If you lose them, then the connective tissue fibers will become continuous across the visceral and parietal layers, and you get an adhesion. You can get inflammation, pericarditis, peritonitis, pleuritis, and then if that leads to break it down of this simple squamous epithelium, you can get the scarring, the fibrous connective tissue, the adhesion. So a pleural adhesion, pericardial adhesion are really not good. Peritoneal adhesion is okay. It's a little less debilitating and uncomfortable. So we're just picturing looking into the purse. You got a thin silk layer lining the purse. That's the surface epithelium. We don't want to damage it. So we see the ventral mesentery is still present on the and connected to the lesser curvature of the stomach. The dorsal mesogastrium is attached to the greater curvature, and this omental bursa keeps dilating to the left laterally, and then it will go inferiorly. And just we also see this twisting of the stomach. A lot of other things happen farther down with the midgut, as we'll see. So if the stomach takes on this adult position, we're going to see, we're predicting an outgrowth into the ventral mesogastrium for the development of the liver, the outgrowth into the dorsal mesogastrium for pancreas, a growth of cells, mesenchymal cells, to form the spleen in the dorsal mesogastrium. Then this continued elongation of the dorsal mesogastrium containing a branch of the celiac trunk, the right gastro-omental artery, and eventually this is going to become a long apron to lay down over all the rest of the intestines and form what's called the greater omentum. So let's try to follow that with a horizontal section through the stomach and the dorsal mesogastrium, or greater, the lesser omental sac in this area, and we are going to follow the development of the greater omentum. So don't confuse the greater omental sac space and the lesser omental sac with the greater omentum. Okay? The lesser omentum is going to be the remnant of the ventral mesogastrium on the lesser curvature of the stomach. The greater omentum is going to be attached to the greater curvature of the stomach. So there's a difference between omental sac and omentum. The omental sac is the space inside the purse. So we're looking through the little constriction of our purse with the purse strings on it, and we see the silk lining, and the space in there is the bursa, the omental bursa. But this whole structure, the dorsal mesogastrium, is going to grow and elongate and then hang down. I don't know if you've ever seen old purses where they had the frills that hung off the end of it. So you're looking into the pouch, see the omental sac, but you really don't see the little frills hanging down the bottom of the purse. Those frills hanging down the bottom of the purse will be analogous to the greater omentum that we're following. This greater omentum is eventually going to form this fold, and it'll come together and fuse together and form the greater omentum, as we'll see in an adult illustration in just a moment. So let's review the shape and position of the stomach primordium changing during this development. First, it runs the long axis of the embryo as a fusiform dilation or dilatation of the foregut. Later, it rotates clockwise, causing its left side to face anteriorly and its right side to face posteriorly. So you are in the midst of a tremendous exercise of anatomy in this development. Right and left side, the left side becomes anterior, right side becomes posterior. The anterior surface becomes the right surface, and the posterior surface becomes the left surface. So you have to put this three-dimensional event into your mind and follow it. The anterior surface becomes the lesser curvature. The lesser curvature faces to the right in the adult organism, even the neonate. The greater curvature of the stomach is originally posterior. It ends up facing the left-hand direction. So this visualization helps you in learning the adult anatomy as well. So during the rotation, the original posterior wall grows faster, and it becomes the greater curvature and moves to the left side, as I said. So the cephalic and caudal ends of the stomach are originally located in the midline, right straight in the midline, the two balloons closing down onto it. And then eventually it kind of gets shifted. So the caudal end, or the cephalic end, is going to be moved to the left, and the caudal part is going to go to the right. So there's going to be a displacement of the top over to the left a little bit, and the bottom is going to get displaced a little bit over to the right. So our stomach is right here, and the esophagus is going to be pushed over to the right-hand side. It'll come through the thoracoabdominal diaphragm, just kind of to the left of the esophagus. So it's going to rotate 90 degrees clockwise on this longitudinal axis. Lesser curvature, ventral will move to the right, greater to the left. Original left becomes front, original right becomes back, or ventral and dorsal, respectively. Cranial and caudal ends change, as we noted. The left vagus nerve on the left surface is going to become end up on the anterior side. The right vagus nerve is going to end up on the posterior side of the stomach. Surgeons going in to do any type of surgery must know all of this anatomy extremely well, of course. So here we see the esophagus coming through the thoracoabdominal diaphragm. It will have been just to the left of the aorta. We see the remnant of the ventral mesentery, the lesser omentum, not the lesser omental sac, the lesser omentum. Right here is the opening into the lesser omental sac, right behind the lesser omentum and behind the stomach. Here's the greater omentum extending down from the purse. So it's an apron that continues down. You see its cut-free margin shown here. You see the walls of the stomach. So the endoderm will have given rise to the epithelial lining of the mucosa, the gastric glands, and then the splenic mesoderm will have given rise to all the other components, the submucosa, and all the smooth muscle layers of the stomach. So we can stop there. We have reached this point of discussion. I'm sorry, I will just continue. I was kind of thrown off by this. Let's just quickly do the mesenteries, the dorsal and the ventral. Since we've seen them already, the dorsal becomes the greater omentum, the ventral the lesser omentum. So let's just quickly go through these adult slides. Here we see the liver and the lesser curvature of the stomach. There's the gallbladder. Here's the transverse colon hiding the greater curvature of the stomach. We see that the greater omentum is going to flop over the transverse colon, fuse onto it, and then extend all the way down into the abdomen. So this is a two-layered structure filled with fat cells, lymphatic cells, immune cells, and vasculature. Here we see it has been removed, and we see that it would have been attached to the greater curvature of the stomach, flapped over the transverse colon, and lie in front of all the small intestine in the greater omental sac. Here we see the lesser curvature of the stomach and a film-like structure extending from it over to the hilus of the liver, where things enter and exit is called the hilus. This is the remnant of the ventral mesentery, the lesser omentum of the stomach. We see an opening right here. This is the omental foramen through which fluid can pass into the lesser omental sac. Here we see the greater omentum retracted rostrally or cranially, pulling up with it the transverse colon, so it has a mesentery. It can move. And we see the small intestine with its mesentery proper. We see the descending colon and the sigmoid colon and the rectum and the urinary bladder. So at this point, we can create a definition. If you can pick up a piece of the intestine and move it, and it has an attachment to the posterior abdominal wall or body wall, then it is a mesenteric structure. It will have a mesentery. The small intestine that we see here that has a mesentery is mesenteric. And this is called the mesentery proper, not the well, there is a mesojejunum, a mesoileum. They do break it down, but we just call it collectively the mesentery proper. The mesentery of the transverse colon is the transverse mesocolon. So there is terminology for each of these. The descending colon, you can't pick up and move it around. It's fixed. It doesn't have a mesentery. We'll talk about how that happens later. The sigmoid colon, you can pick up and move around. It does have a mesentery, the sigmoid mesocolon. So we can make that point. So these mesenteric structures are suspended within the greater omental sac by their mesentery. Here we see a transverse section of the, a parasagittal section of the stomach, the lesser omentum attaching from the lesser curvature to the liver, and the greater omentum coming off, flapping in front of the transverse colon and looping down and back onto itself to be attached again to the transverse colon. So this is the greater omentum within the greater omental sac. The lesser omental sac is behind the lesser omentum and the stomach here. So we will start with this slide on Wednesday, the derivatives of the ventral mesentery. I appreciate your time and attention and hope you all had a good weekend. I'll see you on Wednesday. I'll stay here for just a few moments if there are any questions. I guess we should have finished that slide since it's the last slide of the lecture. But anyway, we'll toss it in at the beginning of the next one.